this video, we will be going over carbohydrates within the biological molecules topic. The important thing to learn in biological molecules is what the monomer is, what the polymer is, and what the bond is called that connects these monomers together. So a polymer is multiple monomers joined together. They are joined by a condensation reaction where two monomers come together and a water molecule is removed, leaving a covalent bond. If you wanted to reverse this and break the covalent bond, this would be the hydrolysis reaction, where you need to add a water molecule. The monomer in carbohydrates is called monosaccharides. They are split into reducing and non-reducing sugars. Reducing sugars can donate their electrons and become reducing agents. This is detected via the Benedict's test, which turns the blue copper sulfate solution into an insoluble brick red copper oxide precipitate. Glucose, fructose, and galactose are reducing sugars. Non-reducing sugars cannot donate their electrons, so to be detected via the Benedict's test, the disaccharides must first be hydrolyzed. Sucrose is an example of a non-reducing sugar. When two monosaccharides join together via a condensation reaction, a glycosidic bond is formed. The resulting molecule is called a disaccharide. Maltose is where two alpha glucose molecules join together. Sucrose is where an alpha glucose and fructose is joined together, and lactose is where an alpha glucose and galactose join together. You can use paper chromatography to identify the type of monosaccharides that are present in a mixture. It is important that you know how to label or draw a chromatography diagram for your exam, as well as explain how it would be done and interpreting a chromatogram. You will need a standard solution with known monosaccharides to use as a reference. As you can see here, they will each separate out to a certain distance. Then you put the unknown solution on the pencil line and compare the distance travelled with the original to identify your monosaccharides. Large monosaccharides will move slower than smaller monosaccharides. Colorimetry can be used to identify the concentration of monosaccharides in a solution. This can be an easy 6 marker in your exam. The first thing you need to do is heat your solution with Benedict's solution, then remove the precipitate that forms to obtain the filtrate. Leaving that aside, make up serial dilutions of standard known concentrations, then calibrate the colorimeter using water and put the red filter on. Then for each of the serial dilutions, record the transmission or absorbance. Then plot a calibration curve of transmission or absorbance on the y-axis and reducing sugar concentration on the x-axis. Finally, measure the transmission or absorbance of the unknown filtrate and read off the concentration using the calibration curve. The more transmission or lower absorbance means more reducing sugar is present. The polymer in carbohydrates is called polysaccharides. Starch and glycogen are storage polysaccharides which are adapted for their function by being compact to store large quantities and insoluble to cause no osmotic effect within cells. Cellulose is another polysaccharide which is the structural component found within plant cell walls. They have a high tensile strength due to their hydrogen bonding and have lignin which fortifies the microfibrils, providing additional strength. For your exam, it is important that you are able to compare the structure of starch, glycogen, and cellulose, as well as be able to relate their structure to their function, as being coiled makes it more compact, or having straight microfibrils provides strength, and branched allows easy access for enzymes to break up the glucose to be used in respiration. I hope you found this video useful, and please give this video a like. If you want to see more videos like this, please subscribe.